Let us begin now with the digestion of proteins. Well, the monomer units for proteins are the amino acids. They are what? They are the amino acids. Now, the saliva or the saliva I told you that is present in the oral cavity, it does not have any enzyme for the digestion of proteins. So, the digestion of proteins does not take place in the oral cavity or the buccal cavity. The digestion of proteins in the stomach, right? In the stomach, the pH of the stomach is quite low, it's acidic. The pH of the stomach is acidic in nature, due to which some proenzymes, they get activated. For example, here we have pepsinogen and prorenin. This pepsinogen and prorenin, they are what? They are proenzymes. They are converted into their active forms by the action of this HCl, or you can say the acidic pH that is there in the stomach. So, the pepsinogen is converted to pepsin and prorenin into renin, right. Now, this pepsin and renin, they are going to help in the digestion of the proteins. For example, proteins in the presence of pepsin are changed into peptones and proteoses. Casein in the presence of renin is converted into paracasein, and then we have paracasein plus calcium, and it binds with calcium, it forms calcium paracasinate, that is a curd like substance. And then we have calcium paracasinate in the presence of pepsin, it is converted again into what? Peptones. So, this is how the digestion of proteins is taking place in the stomach by the action of gastric juice. So, gastric juice has an acidic pH. That is why these proenzymes, that is pepsinogen and prorenin, they get converted into pepsin and renin respectively. And this pepsin and renin, they help in the digestion of proteins. As you can see in these equations, we have proteins converted into peptones plus proteoses in the presence of pepsin. We have casein, right, it is a milk protein. In the presence of renin, it is converted into paracasein, and when this paracasein binds with calcium, it is converted into calcium paracasinate, which is a curd like substance. Then calcium paracasinate in the presence of pepsin is converted into peptones. So, this is how protein digestion takes place in the stomach. Remember that the acidic pH is necessary for the activation of these enzymes. When this pepsin is activated, right, when this pepsinogen is converted into pepsin, now this pepsin can convert this pepsinogen into peptin. So, this pepsin can convert this pepsinogen back into pepsin. So, this type of conversion is known as autocatalytic reaction. It is called autocatalytic reaction. Means just for once pepsinogen needs to be converted into pepsin. After that, when this pepsin is formed, this pepsin is going to convert the rest of pepsinogen into pepsin. This is known as autocatalytic reaction. So, this pepsin and the renin, they are acting as very important wonderful molecules which are going to convert, which are going to lead to the digestion of proteins. Just remember these equations, right. Next, digestion of proteins in the small intestine. Let us discuss first the action of the pancreatic juice. Now, in case of pancreatic juice, we have trypsinogen, chymotrypsinogen, procarboxypeptidase and proelastase. Now, these are the proenzymes which need to be converted into their active forms. So, trypsinogen is converted into trypsin in the presence of enterokinase. Chymotrypsinogen is converted into chymotrypsin in the presence of trypsin. And then we have procarboxypeptidase is converted into carboxypeptidase again in the presence of trypsin. Then proelastase is converted into elastase in the presence of trypsin. So, once this trypsin is activated, this can further lead to the conversions. So, that is why I talked about the autocatalytic reaction. So, this is how the digestion of proteins is there in the small intestine, the action of the pancreatic juice. Now, trypsin actually favors alkaline pH. If we talk about pepsin, pepsin actually works under acidic pH, right, that is why we have written HCl, hydrochloric acid, which is there in the gastric juice. But in case of trypsin, we need an alkaline pH, okay, for the working of this trypsin. Then proteins are converted into dipeptides, peptones into dipeptides, proteoses into dipeptides in the presence of the enzymes, that is trypsin, chymotrypsin, carboxypeptidases. So this is how the conversion of the proteins, peptones and proteoses takes place into dipeptides respectively, right. So you can make out the action of pancreatic juice, that is the digestion of proteins in the small intestine. Then here we have elastin. Elastin is converted into dipeptides in the presence of the enzyme, that is elastase. Then let us talk about the action of the intestinal juice. Peptides are converted into amino acids in the presence of aminopeptidases, and dipeptides are converted into amino acids in the presence of dipeptidases. So this is how the intestinal juice also helps in the digestion of the proteins. You just need to remember the equations, you can elaborate them and explain in your own terms. Here I want to give you one important application of this renin, right. This renin is actually extracted from the calf, and this is used for the curdling of milk. This is artificially used for the curdling of the milk, so that you can say the cheese can be obtained. When you talk about the industrial scale, they actually extract this renin, right, from the stomach of the calf, right, or the cow. Why I am talking about calf? Because the amount of renin is abundant, right, in the baby as compared to the adult one. So this renin is extracted and it is used for the curdling of milk. To obtain what? To obtain cheese. This is the industrial application of this renin. Then just remember that when you are going to explain the digestion of proteins, so just always remember that the digestion of proteins does not take place in the oral cavity, because the saliva does not has any enzyme for the digestion of proteins. So it begins in the stomach and finally in the small intestine, where it is converted into amino acids by the action of intestinal juice.